Hello everyone. I am Alisha Khurana from RPIIT Technical and Medical Campus Karnal. So my friends, today in this lecture we will discuss about electrophoresis. So introduction. Electrophoresis is the movement of scattered particles comparative with a fluid under the influence of uniform electric field. Means when we apply electric field, the ions present into the solution migrate based on their strength into the medium. The electrophoresis is of two types based on their ions. The electrophoresis of positively charged particles, cations, is sometimes called as cataphoresis, and electrophoresis of negatively charged particles, anions, is called as anaphoresis. The electrokinetic phenomena of electrophoresis was discovered for the first time by Russian professors Peter Andrews at Moscow in 1807, who noticed that the application of constant electric field causes the clay particles dispersed in water to migrate. Electrophoresis is the basis for analytical techniques used in the chemistry for separating molecules by size, charge, or binding affinity. So, in electrophoresis, we separate the molecules based on their size, charge, or binding affinity. Now, types of electrophoresis. The main types of electrophoresis in particles are zone electrophoresis and moving boundary electrophoresis. Zone electrophoresis includes paper electrophoresis, cellulose acetate membrane electrophoresis, and gel electrophoresis. Moving boundary electrophoresis includes capillary electrophoresis, isoelectric focusing, and isotachophoresis. Now, in this session, we will discuss about only zone electrophoresis. Zone electrophoresis involves the migration of the charged particles on the supporting media like paper, cellulose, acetate membrane, starch, gel, and polyacrylamide. Components that are separated are distributed into the discrete zones on the supporting media. This supporting media is saturated with buffer solution in a small volume of sample and is being applied as a narrow band. Second one is paper electrophoresis is useful for the separation of small charged molecules such as amino acids, small proteins using a strip of paper. In this we use chromatographic paper. For this purpose, the strip paper that is used must contain at least alpha cellulose at a percent of 95% and should have the slight adsorption capacity. A strip of filter paper is moistened with buffer and the ends of the paper are immersed into the buffer reservoirs containing the electrodes. The samples are spotted in the center of the paper. When a high voltage has been applied, the ends of the strip paper. Application of high voltage causes the less diffusion of small molecules, which in rays giving better resolution, and this takes fewer periods to complete the process. Spots migrate according to their charges. Now, cellulose acetate membrane electrophoresis. The cellulose acetate strips are suspended in the tank of horizontal apparatus. For that, both ends are dipped into the buffer solution. Then the sample is placed at one end and the electric field is applied so that it allows the migration of molecules to their respective poles depending upon their charges. Because the resolution and reproducibility of separation in agarose and polyacrylamide gels are better, cellulose acetate membranes are more often replaced by the gel electrophoresis. See here, we use two medium tanks. In one tank we use cathode and second one in anode. In both the tanks we place buffer solution. Now see here, the sample spot is placed on the top and the bands are separated. These bands are of ions and these are separated according to their strength. Now, gel electrophoresis is used for the separation and analysis of macromolecules like DNA, RNA, and proteins and their fragments based on their size and charge. Molecules of nucleic acid are separated by applying an electric field to move the negatively charged molecules through a matrix of agarose and by other substances. The shorter molecules migrate farther than that of the longer ones and move faster because the shorter molecules migrate more easily through the pores of the gel. This phenomenon is called as the sieving phenomena. Gel electrophoresis is of two types, agarose gel electrophoresis and polyacrylamide gel electrophoresis. So my friends, this is all about zone electrophoresis. In next session, we will discuss about moving boundary electrophoresis. Thank you. Please like, share and subscribe our RPIIT YouTube channel. Thank you.